So today we're talking about going to continue talking about molecular separations. We'll start talking about gel-based separations, electrophoresis, capillary electrophoresis, isoelectric focusing, and other methods like liquid chromatography, molecular sieving, a little bit on gas chromatography too if we have time. I'd like to get through most of the molecular separations today and start getting into particle separations. So the goal of molecular separations is to take a mixture of different molecules that are dissolved in the sample and to separate them out and to be able to quantify each one separately. If you had a mixture of A, B, and C proteins, for example, if you're able to separate those proteins from the mixture, separate them out in time, usually this is done by putting the mixture of chemicals through some type of chromatography or electrophoresis system where it comes out the other end at different times, depending on the rate at which it goes through the separation system. And then there's a quantification step. So one example of that, probably the most well-known example, is gel electrophoresis. And I think this is a good starting point for today. This is something that you've probably, or you may have dealt with in undergraduate or even high school biology labs. Gel electrophoresis is used to separate DNA molecules based on their size and charge. It's also used to separate proteins based on their size and charge. The way it works is shown in this graph here. It's based on the concept of electrophoresis, which we ended up talking about last time. Briefly, like electrophoresis is the movement of molecules due to an electric field. And if the molecule has some sort of charge associated with it. Now, if you want to separate DNA molecules, which is used a lot in genomics types applications, DNA happens to be negatively charged. It has a negatively charged backbone, so that makes it very convenient to do electrophoresis. Proteins often can have a charge on it as well. They have something called an isoelectric point, which is the isoelectric point is a pH value at which they don't have a charge. But as long as you're not at the isoelectric point, the protein will have some sort of charge associated with it. So you can use electrophoresis for separating protein molecules as well. So as we talked about, the mobility of a molecule depends on its size. The smaller the molecule is, the faster, the higher the mobility is. And so this is the size factor that's in the denominator. Larger radius will result in a lower mobility. Smaller radius will result in a higher mobility. And then there's also the charge. The charge is in the numerator. The more charge it has, the faster it moves in response to the field. So this can be used to separate DNA molecules by size. So let's see. So this is what the diagram is showing. There's a well here. This is where the sample is placed. So this liquid sample can be placed right in this region. This blue region represents a hydrogel. Now the hydrogel could be made up of agarose, which is derived from seaweed, or it could be something called polyacrylamide, which is used for protein separations. In both cases, the material acts as a sieving matrix. So the way that we can picture this is that the material has a hydrogel is think about a material that has a whole bunch of like small pores in it. Okay, it's a network of pores within there. Okay, and those pores are they're filled with liquid because it's a hydrogel. It's hydrophilic, so it's kind of like a sponge. Think about it like a sponge. Now that sponge has pores in it, right? And if those pores are on the same size scale or close to the size of the individual molecules, then the molecules actually have to squeeze through those pores. Okay, and this will happen even if the pore is much larger than the molecule. Okay, due to electrostatic interactions, on the surface of the pore. Okay, we'll get into some more details on that when we talk about the anisotropic sieving. But the point is, the molecules are going through these pores. Now, smaller molecules can wiggle through the pores easier than larger molecules. So that's the basic concept. So the movement, the electrophoresis of the molecules in the sieving matrix, the small molecules will move faster through the gel than the larger molecules. The way you get them to move through the gel is by applying a voltage. So the sample is placed at the top of the well here, and the blue area represents the gel. When the current is applied, oh, by the way, these gold dots here represent the gel particles. Okay, and you can see, you can kind of see how the molecules, the DNA molecule, for example, how it moves through the gel. It has to kind of go around these gel particles. This kind of represents what a pore is. The spacing between these particles would be the pore. Now, when the negative voltage is applied here, the DNA migrates from negative to positive. So the electric field is pointing from bottom to top. The DNA is going in the opposite direction, and the DNA starts to migrate. The molecules move at a rate inversely proportional to their size. Now, in the case of DNA, instead of thinking about them as a ball, you can think about them as like a long string. They're a polymer. They're composed of nucleic acids that are strung together along something called a backbone, which is like a phosphate backbone. So think about it more like a string. The length of the string determines the molecular weight of the DNA. The heavier molecules are longer than the smaller molecules. So after some time, what will happen is that the small molecules will have moved through the gel faster than the larger molecules. The similar sized molecules travel through the gel at similar speeds. They end up creating these things called bands. Okay, so this is what an actual gel might look like after labeling. After, DNA on its own, of course, it's invisible to the eye. However, you can label it. You can place molecules in there that attach to the DNA, and those molecules might be radioactive. Okay, so those radioactive, they will give off a radioactive signal which can be detected with a camera that is sensitive to that. That's called a radiography. They can also be incubated with a fluorescent dye which makes them glow a certain color. So these bands are basically groups of molecules that had similar molecular weight, similar length. So with a typical DNA sample, you'll end up getting a series of bands like this, and each band represents one particular size DNA molecule. Okay. The reason you get bands of DNA fragments of specific sizes has to do with how you cut the DNA molecule. Now, just a little bit of background. Okay, DNA is a genetic code that is used by all the cells in your body. The same genetic code exists in all of the cells in your body. Now, DNA can be considered a long string. Okay, and that string consists of a genetic code that's in the form of different base pairs. There's 4 possible base pairs. I wish we had more time to go over what a DNA molecule is and why it's important. But just briefly here. The way that you can think of a DNA molecule is something like this. This portion here is called the phosphate backbone. And these are called the bases. There are 4 bases, which are labeled A, G, C, and T. There are 4 different small molecules that attach to the phosphate backbone of the DNA molecule. The phosphate backbone, by the way, is negatively charged. That's what makes the DNA molecule electrophorese. This sequence of bases is your genetic code. Okay, for most of us humans, our genetic codes are like 99.5 or 99.9% identical. It's that those small differences in genetic code that determine, your skin color, your eye color, whether you're tall or short, and so on. So in a typical, in the human genome, there's 3 billion base pairs. So there's 3 billion of these things in the sequence, and so that's your entire genetic code. There's a lot of interest in figuring out what, if you were to take a sample from my body to figure out what my genetic sequence is. That's called DNA sequencing, and that's a very hot field right now. How can you sequence such a long length of DNA? The way they do it is actually by breaking up a long string of DNA into smaller fragments and then sequencing each fragment. And then computer programs are used to kind of stitch those fragments together and figure out the longer sequence. I wish we had time to talk about that, but we could spend a whole semester talking about DNA sequencing. But one of the methods that are used to analyze DNA is gel electrophoresis. And gel electrophoresis will tell you the size of the DNA fragments that you have in your solution, and it'll tell you the quantity just by looking at these bands. You can see that the bands that are further down in the gel, those represent the smaller DNA fragments. The bands that are earlier on in the gel, or that haven't traveled as far, those represents the larger fragments, larger DNA fragments. And how intense this band is an indication of how much of it you have. If there's a lot of it, then this band will be, it'll be thicker, it'll be more intense. Now, this is what a typical gel setup looks like in a biology lab. Your samples are placed in from the top, and notice that you have multiple lanes. So you're actually handling multiple samples at once. The top of the gel is placed at a negative potential. The bottom end of the gel is placed at a positive potential. And these are what the results look like after labeling. You can see that you have like a whole sequence of different bands. Okay, and the position of these bands tell you the size of the fragment, and the intensity of the band tells you how much of it you have. So that on its own can be used. It's kind of a workhorse technology in a life sciences lab. It's even used in a forensics lab. You know how they take DNA sample from a crime scene and determine, they try to determine whose DNA sample it matches up with. Well, if you take a DNA sample and you subject it to something called a restriction enzyme, which chops up the DNA according to certain patterns, an individual will have a certain set of patterns here, a certain fingerprint. That's called DNA fingerprinting. And if the sample that you had at the crime scene matches up with the fingerprint of the DNA sample at the crime scene matches up with the fingerprint of an individual, then you know you have a match. You can say with some certainty that there's a match. And it's all coming from this type of genomic analysis. So this is gel electrophoresis. So we can miniaturize gel electrophoresis on a chip. So this was called polyacrylamide gel electrophoresis. It's not using agarose, it's using a polyacrylamide gel. Polyacrylamide gel is suited for separating both DNA and proteins. It's a high density gel, meaning like the pore size is a little bit smaller than agarose. So the way that they do gel electrophoresis on chip, at least this particular paper, was to prepare the channels. Okay. Prepare the channels first and fill the channel with a monomer solution. Now this monomer solution has a photoinitiator in it. Okay, so that when you shine ultraviolet light into the channel containing the monomer, you end up polymerizing it. When you polymerize it, now this gel region has become a solid, and this is the region that can be used for your separation. So Mohammed, your project is very similar to this type of technique. So why would you want to miniaturize it? And so I encourage you, when you're thinking about your projects as well, think about why you're miniaturizing a system. If you're trying to do some type of analytical technique, or if you're trying to do some type of other device, think about why the physics scales favorably for your project. Otherwise, there was a point early on when engineers had the ability to miniaturize things using MEMS technologies, using photolithography. So engineers started miniaturizing everything. And in some cases, there's no need, or there was no motivation to actually scale a device down. They just did it because it was cool. It was interesting, right? Oh, we can make this very small. And that in itself has value, of course. But whenever you're doing any type of technique or device, think about the benefits of miniaturization. So in the case of gel electrophoresis, miniaturization allows you to do high-speed separations. If we look at this previous slide here, right, in a large-scale gel, proteins will electrophoresis at a certain rate. Okay? So it will actually take them a long time to traverse through this gel, the larger the gel is. If you're doing the same thing in a microfluidic channel, you can still get the same type of, you can get reasonable separations, but you don't require as much time to do it because your size is smaller. There's less distance for it to migrate. Okay, the challenge is maintaining the same separation resolution after miniaturization. So that's the tricky part of it. Another advantage is that you don't require as much voltages. So the electrophoresis rate is given by the electric field, and the electric field is equal to V divided by the electric field E is equal to the voltage divided by the length of the channel. So if the length of your electrophoresis channel is smaller than a typical gel, you won't require as much voltage to perform the electrophoresis. You can maintain the same electric field with a smaller voltage. This makes things a little bit better because instead of a 10,000 volt supply, you could get by with a 1,000 volt, or maybe even a 100 volt supply. So polyacrylamide gel electrophoresis provides high speed, so 40 seconds instead of 60 minutes. And it also provides high resolution. We'll talk about plates per meter, what that means in a few minutes, or a little bit later in the lecture. It can separate biomolecules like mRNA and proteins by their size. So I mentioned how the gels are photolithographically patterned. The typical protocol might look something like this. You'd fill the sample reservoir. So you fill this reservoir here, and then you load the sample by applying a voltage between the sample and the sample waste. So the sample will go down like this. We'll see a video of this with the capillary electrophoresis stuff, if it doesn't make as much sense now. By applying a voltage between S and SW, the sample will electrophorese down this way, from S to SW, so it'll fill this region of the channel. Oops. Then we separate by applying a voltage between this reservoir and this reservoir. So we take off, we remove the voltage from S and SW after the sample is loaded, and instead we apply a voltage between the waste and the buffer reservoir. Notice that we're applying a positive voltage on this side, 0 volts on this side. So the electric field is pointing from right to left. As a result, the molecules in this region start to migrate out towards the right, and the different size molecules will travel at different speeds. So you can image the proteins going through. If the proteins are fluorescently labeled, you'll be able to see the proteins as they come through. And by looking at the fluorescence signal, the pattern of fluorescence, how dark these regions are, you can get what's called a electropherogram. The electropherogram has time on the x-axis, and on the y-axis it has the amount of the molecule. Chromatograms operate in a similar manner. So these are the types of data, or the results that you get from chemical separations. Time on the x-axis, molecular quantity on the y-axis. So over time you can see that from at time 0 there's nothing coming out of the system. At time 1 the smallest molecule comes out, so you get a little peak there. The next largest molecule comes out after that. So you end up having a series of peaks, and each one of these peaks represents one type of molecule that was in the mixture. So when you see an electropherogram like this, you don't necessarily know what the molecule is, but you know that the molecule came out at a certain time. And that can tell you, that can give you hints about the size of the molecule. All right, so think about how this might be used in a setting. You know, let's say your sample, you know that you have DNA in them, and you get a series of peaks like this. Then you can say that, well, I have a DNA molecule of this length, and I also have a DNA molecule of this length, and so on. All right, so it's giving you some hints as to what is in your sample.
So is there a library where you would compare this against to see what kind of...
Absolutely, yes. There are standards that you can compare them against. So, in fact, that's what's done in whenever you do gel electrophoresis like this. You normally will fill one of the end columns with a reference, and that reference contains a sample of DNA with known lengths. They call them DNA ladders. And if you compare, let's say, for example, this was a ladder. Okay, these are known. This is a known sequence, and the manufacturer will tell you that oh, you know, the 6th band that you see on your gel corresponds to 1100 base pair molecular weight, or 1100 base pair length of the DNA molecule. In your sample, if you see a band that's that is at the same position, then you know that this band is roughly around 1100 base pairs. So this allows you to actually measure the length of the DNA. So the gel picture looks something like this. The electropherogram is just another way to represent the same data. You just see a series of peaks in time rather than a visual picture of light and dark regions. So in addition to gel electrophoresis, there's also capillary electrophoresis. Capillary electrophoresis can be used to also separate DNA, RNA, proteins, even things like metal ions. Let's say you want to analyze your drinking water. You want to find out the ionic content of drinking water. How many of metal ions you have in there, and what type. Electrophoresis can be used for that as well. So capillary electrophoresis is a little bit different from gel electrophoresis. It still uses electric fields to separate molecules, but the difference is that it is done in a capillary instead of a large gel. And the second thing is it doesn't require a gel at all. So in this previous example, where gel electrophoresis was miniaturized in a chip, notice you have a very tiny capillary, okay, but that capillary is filled with the gel. So the gel is what is responsible for helping separate the molecules. The gel is acting as a sieving matrix. In the case of capillary electrophoresis, you actually don't use a gel at all. There's a buffer. There's a buffer inside a capillary, a long capillary. Capillary is just a long tube that has a very narrow radius, like a couple hundred microns, even as little as 25 micrometers. It doesn't require the gel. whatever liquid is in there already, usually some type of buffer. Sometimes polymers are used in there. The material itself, what is in the capillary, acts as a sieving matrix. So capillary electrophoresis is very commonly used in medicine, forensics, environmental chemistry, and so on. The idea behind it, if we look a little bit more closely at these graphs, the first thing you do is inject your sample like this, okay, and let's say this is your capillary. This is just a schematic representation. An actual capillary looks something like this. It's long and narrow. So the sample goes in one way. The different components of your sample go through the capillary at different speeds, and then you have a detector at the end. So as these different bands are, each one of these bands represents molecules of similar size. These are all moving from left to right. They just move through at different rates. So if you have a detector at the output, just basically watching for molecules as they pass by, then you end up getting an electrophoregram. It's time on the x-axis, molecular quantity on the y-axis. So in practice, what's done with the capillary electrophoresis, you have an anode and a cathode, and then you have a long capillary in the middle. This is typically a glass capillary. You can buy these things by the meter. They're made from fused silica. And so the inside of the capillary is glass. And so as we know, if you expose glass to water, it forms those electrical double layer. So when you put a voltage across the capillary, you'll get electroosmotic flow. And that's a key part of capillary electrophoresis. The typical dimensions of the capillary would be something like 20 to 30 micrometers in diameter, all the way up to a couple hundred micrometers in diameter. And the length could be 20 to 100 centimeters typical. So if you have a sample inside the capillary, after you insert your sample, the sample is first inserted into the first few centimeters of the capillary, or the first centimeter of the capillary, or even less. Then you place the two ends of the capillary, one at a positive potential, so you have a beaker that's biased at a certain potential, another beaker that's biased at a different potential. So this creates a high voltage on this side, low voltage on this side. DNA molecules will migrate. They'll migrate towards the positive anode, but you get electroosmotic flow going from the anode to the cathode. So there's sort of a balance between the two. That's what ultimately separates the molecules. Let's look a little bit more closely at that. So the procedure will be to first fill the glass capillary with a separation buffer of known viscosity and known conductivity. All right, so you fill the capillary, then you add a short sample plug on the anode side, so you add it on this side. You apply voltage, and this causes electroosmotic flow towards the cathode. Electroosmotic flow, again, is a key part of electrophoresis. Electroosmotic flow goes from positive to negative. So this is the inside of the glass capillary. Remember that it becomes negatively charged. You build up a layer of positive ions next to that, next to the surface. And when you place a voltage across the length of the capillary, these positive charges start to move in the direction of the electric field, and they drag the rest of the fluid with it. So electroosmotic flow goes from positive to negative. Now suppose you have DNA molecules in there. So DNA molecule would be considered an anion because it's negatively charged. Now in this case, the electroosmotic flow is going to cause the DNA molecules to move from left to right. However, the electrophoresis is going to cause the DNA molecules to go from right to left. Right, so you get this opposing, you get two opposing effects. For cations, you have electroosmotic flow going from left to right, And then the electrophoresis also causes the molecules to go from left to right. So you even get a stronger movement from left to right. And then if you have neutral atoms, then those are subjected only to the electroosmotic flow. So notice that the electroosmotic flow velocity is the same regardless of what type of ion you have. Okay. The electroosmotic flows, that's moving the ions from anode to cathode all at the same rate. However, the electrophoresis causes these things to move at different speeds. So the anions would come out last, the cations would come out first, and even within the anions here, this rate of electrophoresis depends not only on it depends on the charge, and also depends on the size of the molecule. So even if you just had anions in there, you could separate them out by size and charge. And that's what's done with DNA electrophoresis. So these different molecules come out at different times, and the detector monitors them. The DNA molecules would be, you can monitor them using, if they're fluorescently labeled, you can monitor them with a fluorescent detector. It is even possible to monitor certain molecules with just an absorbance detector, although it can be difficult because the path length is very short. If you're trying to optically detect a molecule, it doesn't work very well at the small scale. That's something we'll talk about more in the detection module. All right, so does this make sense? Everyone kind of see how you can separate molecules with capillary electrophoresis? So this type of thing has been done for a long time. The capillary electrophoresis systems have been available for decades. They were, rather large systems, and they were ultimately miniaturized. But it's worth it to see what some of the prior generations of the systems look like. So this is an early system, and this is a somewhat more modern system. These types of systems are actually still sold commercially. These systems allow you to inject a sample. It basically allows you to do this procedure that we showed here, but that does it in an automated manner. Typical pressures for injecting the sample would be, a couple hundred millibar. The voltage that you put across the capillary, okay, going from here to here, is like 20 to 30 kilovolts. That's a lot of voltage. It's a very high voltage. The wall outlets give you, 120 volts. This is like 30 kilovolts, 30,000 volts. So, and one of the disadvantages of traditional capillary electrophoresis is that you do need a very high voltage supply in order to run it. This is because the length of the capillary is typically 30 to 100 centimeters in length, and as I told you earlier, electrophoresis depends on electric field. Electric field is the voltage divided by the length of the capillary. E is equal to V over L. So if your capillary is very long, you're going to need a very large voltage across it to drive the electric field. Because you have a long capillary, it also takes the molecules a long time to go through there. So the separation time is minutes to tens of minutes. And the sensitivities, are somewhere on the order of 1 micromolar. Okay, and it also has these integrated detectors. These are commercial systems, prior generations of CE. So in the early 90s, early to mid 90s, the Harrison and Manns reported some capillary electrophoresis systems that were done on a microfluidic chip. Now, this was an important time because this is really the beginning of the microfluidics field. The modern microfluidics field that we think about with microfabrication techniques, microfluidic chips. This was really the beginning of it. In fact, I think it was Andreas Manns was one of the guys who coined the term like lab on a chip or micrototal analysis systems. So this field, you can think about it, is relatively new. Relatively speaking, it's not that old. It's probably like only 20 years old. There's been a lot of development since some of these early landmark papers. So the first demonstration of microfluidics was with the capillary electrophoresis on a chip. And there are some advantages of miniaturization. First of all, you're just as is the case with gel electrophoresis. By the way, that gel electrophoresis paper was published sometime in the 2000s. This is the earliest microfluidics work. The advantage of the system is that you can do fast separations. Whereas the traditional capillary electrophoresis systems may take tens of minutes, this capillary electrophoresis system on a chip can do separations within tens of seconds. So it's a huge difference in terms of analysis speed. So you can imagine why a forensics person or a life scientist would be interested in that if they can do their analyses much faster. So the CE microfluidic chips were cross junctions. The length of these things were 1 to 10 centimeters, and the cross section, meaning the radius of the channel or the width and the height of the channel, were around 10 to 30 micrometers. And they were made in glass because you need glass to get electroosmotic flow. So the material that you use is very important. And the procedure is similar. You do electrokinetic injections. Well, I shouldn't say similar. I mean, you still use electric fields to do the injection, but it's actually done in a pretty interesting way. Let me describe it, and then we'll watch this movie. So the procedure, so you have a cross chip, a cross junction like this. Okay. The vertical dimension is used to load the sample. Okay, so you start off with the sample up here, and when you apply voltage between the top and the bottom, the sample is loaded vertically into the sample loading channel. And then the second step is you apply voltage between the left side and the right side, and that causes whatever sample that you had right at the middle of the junction, that sample gets pushed from left to right, and whatever is in that sample, those molecules come out at different speeds, and you do your separation. Now let's look at this video. By the way, this video is from Andreas Manz's YouTube channel. Andreas Manz was one of the early pioneers in this area who did CE on a chip. Let me slow down here. So what's happening at the beginning is that we're doing an electrokinetic injection. We place a voltage between the top and the bottom, and due to electroosmotic flow, the sample gets loaded in this loading channel. Next, what you'll see is that you're going to remove the voltage from the top and the bottom, and instead you'll apply voltage from left to right. And you see whenever we did that, right when that happened, this little sample plug starts to electrophorese from left to right. And you see how it separates into two separate bands? That's because there's two components in that sample of different sizes. The molecules in that sample are different sizes. What we're doing is that they're separating two fluorescent dyes, and those fluorescent dyes have different sizes. So the smaller molecule travels faster. So this portion here is called a sample plug. And as the sample plug travels downstream, it separates into bands. If you had a detector that was sitting here on the right side and just watching what comes by, you'll get a series of peaks. So this is what an electropherogram might look like. They were separating amino acids in this particular example, and those amino acids were fluorescently labeled so that you could see them moving through the channel. So the buffer, again, the buffer is loaded on this side. The whole chip is first filled with buffer. Sample is loaded from top to bottom, and then you apply voltage from the buffer to the waste to get the sample to go to the right and separate out. Since this was the famous paper published in the most famous research journal, Science. It's published in 1993. And since then, there have been hundreds of papers, probably thousands of papers written on capillary electrophoresis on a chip. And in fact, this technology has actually been commercialized by two companies if not more. So this can get 75 theoretical plates in 15 seconds or 600 plates in 4 seconds. This is similar to the separation resolution of capillary electrophoresis, but it's 1 to 2 orders of magnitude faster. I think I left the video on. Let's turn it off. We'll talk about plates a little bit later. Plates is a quantitative metric for separation efficiency. We'll talk about that in a little bit later. So they're saying that you can get similar separation efficiencies to conventional capillary electrophoresis, but it's 1 to 2 orders of magnitude, 10 to 100 times faster. So there's a great interest from industry to commercialize this technology, and indeed it has been. So when we think about miniaturization, another thing we can think about, well, if we can make the device small, instead of making just one channel, why don't we make hundreds of channels? Now there has to be a reason for doing that. So the application is called a capillary array electrophoresis. When you want to do high throughput genetic analysis, meaning you want to be able to analyze many samples simultaneously, or you want to analyze many samples at once, this is where capillary array electrophoresis comes in handy. You still have a single voltage source, but that voltage source is being applied to many, many channels simultaneously. In a sense, it's similar to, in a gel, you have all these lanes of the gel that are being analyzed simultaneously. You just have a single voltage source between the top and the bottom of the gel. So with capillary electrophoresis, you could have, for example, this was showing 384 different separation channels. The voltage is placed from the inside of this disk to the outside of the disk. And the separation channels are arranged in a radial format. So at each one of these spokes represents one capillary electrophoresis channel. So you could do all 384 experiments at once. Now, the trick with that, the tricky thing is that you have to detect all the channels at once. It's easy to apply a voltage between the two ends of all the channels. Yeah, applying the voltage is easy, and the capillary electrophoresis will happen in all the channels simultaneously. The tricky thing is monitoring all 384 channels at once, doing the detection on all 384 channels. So the way that they were able to do this is by using a sort of a rotary setup. This rotary setup, it could spin this objective in a circular manner, and as this thing was spun around, this thing was just going through each of these radial channels one by one and looking at the fluorescent signal coming from those channels. So this disc could spin relatively quickly, so they were able to monitor all 384 channels relatively quickly, almost pseudo-simultaneously. Now, this is the optical setup that's used to detect fluorescence. We'll talk more about that in the detector module, in the detection module. You require some optics. You have to actually illuminate the sample with a certain color light. You have to illuminate it with blue light. That's what's happening from the laser here. And then you have to detect the green light being emitted from the sample. So it turns out the blue light goes through this optical path, and whatever fluorescence is emitted goes back through here, but it goes back this way, and it's detected at a light detector on this end. So they showed these results that you can separate 384 channels at once. You got some very nice results there.
So the rotation doesn't impact your separation?
No, that's a good question. Yeah, that's a really good question. The rotation of the device in this case, it didn't affect the separation. If you are spinning at very high rates, and if you're dealing with larger channels, then you could get centrifugal flow, which would cause, the centrifugal flow would cause a flow in the channels. You obviously don't want that. With capillary electrophoresis, you only want electroosmotic flow. In order to prevent the centrifugal flow, they filled the channels with polyacrylamide gel. If you have gel in the channel, then there's not going to be much flow. Yes, I'm glad you brought that up. So yeah, the channels are 8 centimeters long, and they're filled with the polyacrylamide gel. Microchip electrophoresis has been commercialized, as I mentioned, by Perkin-Elmer. It used to be called Caliper. And Caliper Technologies was founded in the 90s by Michael Ramsey, a researcher. I believe he's now at the University of North Carolina. And he was one of the early pioneers in this field. Caliper was recently bought out by Perkin-Elmer. And this is an example of some of their chips. Agilent Technologies, both these companies are based out in California. Agilent Technologies also has a microfluidic chip used for capillary electrophoresis and DNA separations. So these products, they measure DNA and RNA fragment size and also their concentration. You can deal with DNA sizes ranging from 12 to 1,200 base pairs, and you can do separations in 30 to 60 seconds. Again, traditional electrophoresis with gels takes tens of minutes. So there's a huge advantage here. The typical protocol, this is the protocol for the Perkin-Elmer chip. It's called a SIPR chip. You can see that this is a nice schematic that actually shows what the microfluidic channels look like, the CE channels. But one of the practical challenges in doing these kinds of experiments is how are you going to actually load your sample into the chip? Sample loading, I mean, it seems like a rather mundane topic, but it turns out those little details make a huge difference in how useful a particular product is. So Caliper had something called the SIPR chip, where they had their microfluidic chip sitting on this cartridge here, but they just had a capillary sticking out of it. And the capillary pulls liquid in. Remember, we talked about capillarity last module. Through surface tension, it pulls the liquid directly into the chip. So it turned out this thing, which is called a SIPR, turned out to be very useful in transferring liquids from the traditional microplate formats, those trays, the plastic trays that have the experiments in them. And this, by just aligning this tube over one of those wells, you could pull your sample into the chip, and you can pull a prescribed amount of it into the chip. So it really made things much easier in terms of laboratory automation. Other than that, things are similar to what we talked about earlier. The sample is mixed with a dye to label it, to make it fluorescent. The electrophoretic separation is done in the CE channel. The detection is using a fluorescent optical detector, similar to what we saw on the last slide. And then there's software for analyzing the electropherogram. So this is the type of data that these chips might return. They have an electropherogram. So this would be the time axis, and then the fluorescence intensity on the y-axis. So they compete with things like gel electrophoresis, agarose and polyacrylamide gel. So a researcher could use a gel that looks something like this, where you'd have these bands in there, or you could use this automated instrument that would presumably, at least according to their marketing, that has much sharper bands than the acrylamide gel. Notice that in the gel you have some of these effects where you have this sort of a smile. They call these kind of a smile effect. So instead of having straight bends, you have a little bit lower speeds around the edges. This is due to the additional, first of all, there's non-uniformities in the electric field, and second of all, the fact that the molecules are close to the walls of the channel, they travel slower. Because remember, flow velocity for everything is slower near the walls of the channel. So you end up getting this, instead of a straight band, something that looks like this. In electroosmotic flow, remember, electroosmotic flow is originating from the walls of the channel. So you get that flat flow profile in electroosmotic flow. So for that reason, you can end up getting much sharper peaks when you're doing this with capillary electrophoresis. So here are some examples of what you can do with, when you separate and quantify a mixture of chemicals. Let's say you're separating DNA and quantifying it. This can be used to analyze fragments, molecular footprinting, as I mentioned earlier, for forensic applications. It can be used in the analysis of food. So you've all heard of GMOs, genetically modified organisms. You can actually take a seed from there and use a restriction enzyme to chop the DNA up into smaller fragments and put that into a DNA analyzer. And based on these fragment sizes or the electrophoregram that you see, you can actually determine whether it was a genetically modified organism or whether it was a wild-type organism. It can be used for cancer detection because cancer cells may have a different genetic fingerprint than regular cells. It can be used for gene expression. That's something that we haven't had much time to talk about. It can be used in the preparation of DNA sequencing libraries. So this is one of the steps that can be used in the process of DNA sequencing, which is finding the codes, finding the actual bases in the DNA. In terms of protein analysis, you can measure the amount of proteins in a certain sample, protein expression, purifying proteins from samples. This can also be used for food if you want to find out how much protein or how much of a certain type of protein is in a food. You can use it for detecting antibodies too. Antibodies are molecules that attach to proteins, and they're important in your immune system. And then there's other industrial uses as well in agriculture, drug development, genomics, proteomics. So this is a very workhorse type technology. Any type of life sciences lab will have some type of capillary electrophoresis system in there. And thanks to miniaturization and BioMEMS technologies, these systems are much faster now than they used to be. So let's talk a little bit about separation efficiency. So an electrophoregram looks something like this, right? You have these peaks that come out at the end of the column, and if we want to quantify separation efficiency, just as a qualitative thing, let me say that this would be an example of a bad separation. In a bad separation, you want to separate out all your molecules in time. So a bad separation would be something like this, where you have two peaks that sort of overlap. Let's say this was component A and this was component B. They didn't separate completely in the electrophoresis device. So this is also called co-eluting, where two different size molecules, instead of coming out at different times, they come out at roughly the same time. So now you don't have a very good separation. An example of a good separation is something that looks like this, where you have two distinct peaks. A came out at a certain time, and then B came out at a different time. This would be a good separation. So if we want to quantify the ability of a system to do good separations, ideally what you'd like is that you'd like these peaks to be very narrow and very short in time, tall and narrow. So let's say an even better separation might look something like this, where you have a very narrow peak. Now the reason why you want narrow peaks is so that you can fit more samples or more components within your chromatogram. You know, the narrower the peaks are, the more components you could potentially separate and analyze. So a really good separation process will have very narrow peaks, and that allows you to have a high peak capacity, the number of peaks that you can accommodate in the chromatogram. Yep.
It depends on the length of the channel, length of the, because if you have a long time, we can separate it.
Absolutely, yeah. It does depend on the length of the channel. Yeah, because you need time for the molecules to be able to separate out into bands. There's a lot of factors that determine separation efficiency, but the length of the channel is one of them. In gas chromatography, for example, the longer the channel is, the more efficient the separation will be. The way that you can quantify this in terms of numbers is by something called a plate count. And the plate count is related to the retention time here, and it's also related to the full width half max. So a very narrow peak would be something with a very small FWHM. The formula for plate count looks something like this. There's a historical origins for this formula. We're not going to get into that. But notice that the full width half max appears in the denominator, and the retention time, TR, is represented in the numerator. It is expected that the longer a band is in your column, that band may spread out a little bit due to the diffusion of molecules within the column. But that's why this is expressed as a ratio. If your full width half max is very small, then the plate count will be very high. A high plate count means you have a very good separation efficiency. And you can see that it's inversely proportional to the square of the peak width, or the full width half max. So the peak capacity is the total number of peaks we can obtain without overlap. And for a one-dimensional separation, this is the formula for the peak capacity. Okay, notice that it is the full width half max is on the bottom, and the time of the separation is on the top. So it's basically just showing this diagram here. If you had a whole bunch of peaks and each one of these peaks had a full width half max, how many could you fit into your chromatogram? So this scales as the square root of n. So the higher the plate count, the more peaks you will be able to accommodate. So the factors affecting the plate count, as Hamid said, the length of the separation column is one of the factors. Longer columns are more efficient, but they're slower. They take a longer time to complete the analysis. It also depends on the molecular properties like diffusion, depends on temperature, depends on the presence of flow, like Taylor dispersion. If you're doing separation inside a microchannel, if you have Taylor dispersion, that will affect your separation efficiency. And it also depends on the type of separation process. We've been talking about gel and capillary electrophoresis thus far. So going back to this slide earlier, 75,000 plates in 15 seconds is pretty it's pretty remarkable, because it's similar to conventional CE, except the time of separation is much faster. So continuous flow electrophoresis eliminates the sample loading step. This is another way to do electrophoresis. So let's look at the difference here. So conventional electrophoresis looks something like that. Conventional electrophoresis looks something like this, where you load your sample into the channel, and then you have a separation channel, and you have a detector out here. So you get an electrophoregram with time on the x-axis, and the signal is on the y-axis. In continuous flow electrophoresis, it's a little bit different. Instead, what you do is you load your sample into a channel, and that channel has laminar flow. The field that you put in here is orthogonal to the direction of flow. That is the key difference. In conventional electrophoresis, all the molecules are moving in this speed due to the electroosmotic flow, and the electrophoresis is parallel to the same direction. Here, the sample is flowing from left to right due to a pressure-driven flow, and then you have an electric field going in the orthogonal direction. Right, so this requires a pressure-driven flow. So the force field is causing the molecules to move down orthogonally. So as a result, if you have a mixture of molecules that went in here, if you didn't have any electric field, all the molecules would just go straight across the channel due to laminar flow, right? We've seen that before. You know, the parallel streams, they flow parallel to one another. So in the absence of the field, the entire sample would just flow straight through the channel like this. But if you add an electric field, the electrophoresis is going to cause the molecules to kind of also move down at different rates. So as a result, the small molecules, the small and highly charged molecules will move down to a lower part of the channel, and the larger, less charged molecules will migrate slower. So you'll end up with these different bands at the end of the channel. So instead of having your electropherogram, instead of being time versus the signal, you'll have position versus the signal. Your detector actually has to scan across like this, across the length of the channel. So the definition of the continuous flow electrophoresis, the direction of the electric field is orthogonal to the direction of flow. One of the potential advantages of this technique is that the heat generated by Joule heating is dissipated quickly, so this allows you to do higher voltages, and thus you can get faster separations this way. The fabrication of these devices is a little bit tricky though. You can see that a free flow electrophoresis system looks something like this. So you have to have some sample inlets here, like so. And then you have to apply an orthogonal electric field across here like this. Now in order to apply that electric field, you have to put electrodes inside this portion of the chip, or put wires, have this connected through external wires. Now that can generate bubbles in the flow. So whenever you put a voltage across something, you can generate bubbles. This is a problem with CE too, so you have to be careful with that. But the purpose of these side channel arrays is to allow the electric field to pass through there, so you still have your electric field going through this device, but it prevents any bubbles from going in there. And it also prevents any hydrodynamic flow outside this way, because you would like all your hydrodynamic flow to go through the length of the channel. So this is your sample inlet. Your sample comes in this way, and then it moves across in time. These are examples. So the sample entered the system on the left here. This is with no electric field. The sample just goes straight down. And when you apply an electric field, you can see that the charged components start to migrate across this way. So the x-axis is the distance across the separation bed here, and the y-axis is the fluorescence intensity, so you can nicely visualize these molecules separating out in space rather than time. Questions? So from a philosophical perspective, continuous flow electrophoresis is for continuous flow separations. Notice that this is very different from a batch flow process. In the capillary electrophoresis system, which we saw, you're loading your sample and then you're doing a separation. You're loading a sample, you're doing a separation. So it's a batch process. You're doing one sample at a time. Now, let's say you want to analyze running water. Right, with running water, you're not going to. If the water is flowing, if you could continuously analyze the water, that's a nice way to be able to analyze a sample. So batch analysis versus continuous analysis. The continuous flow electrophoresis systems allows you to do this types of continuous analysis. An example of where you might want to use continuous analysis, I mentioned water. Let's say you want to continuously measure the ionic content or the ion concentrations in your drinking water. You could design a continuous flow electrophoresis system to do something like that. So another analytical technique is isoelectric focusing. It's called IEF for short. So you can see that in this class we're just kind of like rolling through, kind of giving you an overview of the different analytical techniques. And half of the stuff we're talking about is, just a conventional technology, and then half of it's, going into the microscale implementations of these techniques. But having a broad overview, I think, will be helpful to you, because this is really where these types of analytical techniques are really where the commercial applications of lab-on-a-chip systems are, at least right now. So isoelectric focusing is used to separate proteins by their isoelectric point. We want to be able to separate molecules based on many of their properties. Okay? Electrophoresis allows us to separate molecules on the basis of their charge and by their size. Those are some of the most important properties of molecules, but it's not the only one. Okay? Let's say two molecules have the similar size and similar charge. We can't separate them out anymore, right? Let's say two proteins have similar charge and similar size. There's no way we can isolate them. So the more separation techniques we have that allows us to separate things by different properties, that will give us more, improve our toolbox, for being able to separate different biomolecular components. So this is a technique which allows you to separate things by something called the isoelectric point. The isoelectric point is the pH at which a protein has no charge. You may remember this concept from chemistry that Nick remembers, I'm sure, right? The fact that like molecules can have different charges at different pHs. So the isoelectric point is a pH where the protein has no charge. This is a schematic of it. It's at low pH when it's acidic, most proteins have a positive charge, and at high pH, most proteins have acquire a negative charge on their surface. So if you were to place a voltage across here, well, let's back up for a second. The key part of isoelectric focusing is to separate proteins by their isoelectric point. The key part is the pH gradient. So there's a gel here, a gel that has different pHs at different points. There's a pH gradient that's established across the gel. So this end of the gel is acidic, and this end of the gel is alkaline. And the pH goes from 0 to 14 as we go from left to right. Now there's the ways that you can create those type of gels is by using something called carrier amphilytes. They're mixtures of molecules which form pH gradients in the presence of electric fields. So if you just had a gel, and the gel was doused with it was doused with the carrier amphilytes. So this mixture of molecules, if you apply an electric field over them, they'll actually create a pH gradient. We're not going to go into the details of how that works, but just know that it does. Another way you can create a pH gradient is by using immobilized molecules. This is done by attaching immobilines to the polyacrylamide gel during casting. And this type of approach it's generally more stable, higher resolution. Gels that have immobilized IPG gradients, you can buy them. They're available from many manufacturers. So this is what's often used when you want to do isoelectric focusing. If you want to have a little bit more control over things, then you can use these carrier ampholytes. So once you have this pH gradient, let's go back here. You place a positive voltage across the gel, okay, and assume that you have a pH gradient here, going from 0 to 14. So there are going to be certain proteins that have, if the proteins start off on this side, they'll start off with a negative charge, and they'll migrate from right to left. If proteins are on the acidic side of things, they'll generally have a positive charge, and they'll move from left to right. The proteins will basically continue migrating due to electrophoresis until they reach their isoelectric point. Remember, electrophoresis requires the molecule to be charged. So as long as the molecule is charged, it's going to continue electrophoresing. This molecule, for example, goes from left to right, and once it reaches the point in the gel, its isoelectric point, the pH at which its charge disappears, or its charge is balanced, at that point it stops electrophoresing. So you could keep the electric field going on indefinitely. The molecule will eventually stop at a certain location in the gel. And molecules at the similar isoelectric points will form bands in the gel, similar to electrophoresis. So the proteins stop migrating because they reach their isoelectric point at a certain level. These are just some formulas describing the separation, resolution, and peak capacity of isoelectric focusing. This also shows the isoelectric points of some common proteins like egg albumin, hemoglobin, and so on. Right, so this is a combination of electrical techniques and chemistry that are, and pH chemistry techniques that are used to separate molecules. Isoelectric focusing has been miniaturized, and it's been miniaturized using microscale IPGs, the immobilized pH gradients. So they have these polymers that have these gels that have the IPGs immobilized on them, and you apply a voltage between the two sides, and you can see that these molecules will go to their isoelectric points. In this case, each of the molecules is fluorescently labeled so that you can see them. This achieves a resolution of 0.04 in terms of PI, the resolution of the isoelectric point that you can measure. A peak capacity of 78. So the advantages of miniaturization here is that, again, it's a time thing. The smaller the device is, the faster the proteins migrate. So in this case, experiment took 20 minutes rather than 18 hours. So the interesting thing is that you can get those faster analysis times without sacrificing the resolution. Isoelectric focusing can also be taken to the free flow format. So this is the analogy to the continuous or free flow electrophoresis. So in this case, the sample comes in at the top of the chip, but it comes in at the center inlet. There's pressure-driven flow going from top to bottom here, and the electric field is going from left to right. So the key thing in here is that the buffer in this channel, okay, the aqueous solution that's in this channel has carrier ampholytes. So when you place an electric field across the width of the channel, this creates a pH gradient in the channel. Right? So these samples are they're going to move in the direction of pressure-driven flow. Of course, they're going to move from top to bottom, but they're going to migrate laterally simultaneously. They're going to also migrate laterally due to this due to electrophoresis. They'll stop migrating once they get to their isoelectric point. And this is what the system might look like. The sample, this is free flow electrophoresis. This is free flow isoelectric focusing. Your sample can actually come in from all of the inlets in the channel, and if the channel is long enough, they will get focused to certain outlets. Okay, the carrier ampholyte is being applied to this system here. Okay. This is free flow electrophoresis. This is free flow isoelectric focusing. So imagine just being able to take a sample, putting it into the chip, and you have all the separated components just coming out at different outlets. That's pretty cool. A lot of microfluidics is focused on separating biological mixtures like this. So this was shown that you can separate not only molecules but also like particles and subcellular organelles by their isoelectric points. Isoelectric points they apply to particles as well as molecules. There's some trade-offs with this device, things like Taylor dispersion because you have pressure-driven flow, stability of the pH gradients, and so on. You know, all in all, this is a pretty interesting technique. So I'm going back to this slide. This is kind of an old slide, but it's trying to give you a perspective in terms of, you know, what is the life scientist doing? Where are these analytical techniques being used? You know, one important application is this thing called proteomics. I don't know if any of you have heard this buzzword before, but proteomics is the study of proteins. You know, how much proteins are a certain cell secreting. If I were to take a blood sample, in your blood you have, you know, thousands, more than 10,000 different proteins in your blood. And the levels of those proteins actually tell you about the state of your health. That's why you have blood tests at the doctor's office. So if there is a way to take a blood sample, for example, and separate out all the different proteins and somehow measure the different quantities of the different proteins, that's one, you know, that's in some ways a holy grail of the proteomics field, if you could do something like that. And to be able to do the same type of technique not only to blood samples, but also other biological or environmental samples. So the workflow that's done right now is they do two-dimensional gel electrophoresis. We'll talk about this in just a second. This is a two-dimensional method to separate out proteins based on two different properties, based on size and charge, also the isoelectric point. This would go into, you can do an image analysis of this gel. This 2D gel like separates out all the gel, so each one of these spots that you see here is a certain type of protein. You can quantify it just by looking at the intensity of each one of these spots, or if there's a certain protein that you want to analyze, you can pick it out with a robot and pick out this spot, for example. You can do further analysis on it. The further analysis would involve digesting the protein with a protease and breaking it up into its amino acids, and then performing something called mass spectrometry to basically identify what that protein is. We haven't talked about mass spectrometry, and we probably won't get a chance to this semester, but mass spectrometry is a way to analyze the components. It breaks up a molecule into constituent pieces and is able to identify each one of those pieces. In the case of proteins, protein can be fragmented into the building blocks called peptides, and the mass spec can actually tell you what those peptides are and how much of them there are. So 2D gel electrophoresis looks something like this. And again, this is not a BioMEMS technique. It's a traditional technique. We'll get into the miniaturized versions of this later. 2D gel electrophoresis, the reason why it came about is because you have a certain peak capacity with one-dimensional separations. If you do electrophoresis, you'll get a series of peaks on a chromatogram, right? But you can only fit a certain number of peaks on there, right? If you think about this another way, let's say you were to try to separate out all the proteins in a blood sample. Let's say there's 15,000 proteins in there. There's definitely going to be some proteins that have the same size and charge, or similar size and charge. So those proteins will come out together. They'll co-elute. So you won't be able to analyze them and isolate them separately. This is where two-dimensional techniques come in. You actually separate in 2 dimensions. You separate in the x dimension based on one property, and you separate in the y dimension based on a different property. So in the case of the this type of separation, proteins are first separated using isoelectric focusing in the x dimension, and then they're separated by molecular weight using traditional gel electrophoresis in the y dimension. Then they're stained, and after you stain them, or basically a labeling, you'll see all these different dark spots, and those dark spots represent the amount of proteins that you have. Okay, this looks like just a messy paper, but this was a very exciting result where they first showed that the position of these spots actually and the intensity of those spots tells you how much of each protein you have in a sample. It's early but very exciting work. In a traditional, in a typical CE separation, you might be able to separate less than 100 different proteins. And in two-dimensional separations, you can separate more than 1,000. So there really is a big motivation for being able to do two-dimensional separations. 2D techniques can resolve hundreds to thousands of different proteins. There's a 10 to the 7th dynamic range in protein concentration that can be detected by this. And basically, the darker the spot is, the more protein there is. Now, 10 to the 7th dynamic range, that's a very large range. 7 orders of magnitude difference in concentration you'll be able to detect. So this was started in the 70s and mid-70s, and now it's pretty much a more commonplace technique. You can buy two-dimensional gels now and do these separation experiments. The gels have an immobilized pH gradient that has those IPGs on them for forming the pH gradient, and that's in the x dimension, and in the y dimension it looks like a traditional gel electrophoresis, and that requires high voltages. They use polyacrylamide gels because that gives good separation. You can kind of see how where something like this would be useful. This is a certain cell type that they were experimenting on, and they took a bunch of those cells, they digested them, and they subjected them to this two-dimensional gel electrophoresis, and they found that a certain pattern of protein spots. And after those proteins were transformed or transfected, that changes the behavior of the cell. It changes what the cell is secreting and producing, so you can actually see the differences in the different proteins that where their behavior changed. For example, this protein was being highly expressed by the normal cells, but the transfected cells that reduced the amount of production of this protein. So being able to see the entire change in just one shot is a very useful technique. So the microfluidics folks have attempted to do two-dimensional separations on a chip. So this is an example of where a 2D polyacrylamide gel electrophoresis on a chip. I'm sorry. No. This is free solution. This doesn't have a gel in it. I'm sorry. Now let me make a correction to that. So they perform isoelectric focusing in the X dimension and then capillary electrophoresis in the Y dimension. Now notice that they're not using a big slab of gel. They're using just a simple cross channel like this. So it's kind of interesting how they do this. The procedure is they first perform isoelectric focusing along the length of this channel between A and C. And then, so they start off by focusing in the X dimension using isoelectric focusing. So you form these different bands here. This is the junction here. The next step is they move one of the bands. Let's say they want to analyze this particular band. They move that band using electroosmotic flow. They move it into the intersection area. Then they apply a field in the Y direction. So they remove the field in the X direction. They apply it in the Y direction so that they can do a traditional capillary electrophoresis separation in the Y dimension. And then after one band is fully analyzed, they move on to the next one. So you can see this is band N, and this is the next band, N-1. They first subject this band to the second dimensional separation, and then they move the next one in line to the intersections to do the next one. So all the bands that were formed in the first separation are then analyzed in the second dimension of separation. So this is called a peak parking. So two-dimensional separations gave them a high peak capacity as well, 1300. So the peak capacity is a product of the peak capacity in the first dimension, 130, and the peak capacity in the second dimension, which was 10. So they have a better peak capacity in the first dimension. Isoelectric focusing gives you better resolution than traditional capillary electrophoretic separations. So focusing steps are actually much better. So they give you a higher peak capacity. The second peak capacity is only 10. But when you combine the two together, this is the advantage of 2D separations. The peak capacity in a 2D separation is the product of these two. So 130 times 10. So it greatly improves your the ability to separate complex mixtures. Because they didn't use a gel, they used free solution, and they did this in a microfluidic chip, they had fast analysis times, less than 1 minute compared to it would take hours to do this on a traditional gel. So let's see, I think over the last 10 minutes we'll just finish up this stuff. The next thing to talk about is liquid chromatography. So we've talked about electrophoresis, we've talked about the many variations of electrophoresis. Now we could talk about chromatography. Electrophoresis was a separation of molecules by size and charge, and they're being separated in the presence of an electric field. Chromatography is a slightly different concept. It's also used to separate a mixture of chemicals, but the chemicals are placed into a column that contains a stationary phase. For example, let's say you have a metal column, so a metal tube, and in that tube you have it filled up with some sort of slurry. Okay, slurry or beads, and those beads have certain properties, certain retaining properties. The mixture of chemicals that goes into the column will travel through the column at different speeds due to the interaction of the molecules with the stationary phase. So I gave you one example. A stationary phase could be a column of packed beads, a bunch of spheres, small spheres that are packed into the column. And those spheres may retain, for example, one type of separation is called reverse phase chromatography. Reverse phase chromatography retains hydrophobic components more than hydrophilic ones. So if you have hydrophobic proteins, they will go through the column slower than the hydrophilic proteins. So this is, the chromatography allows you to separate based on properties of the hydrophilicity of the proteins in this case. There are also chromatography columns that allow you to separate by size, by charge. There's something called anion exchange chromatography columns that allow you to separate by charge. Size exclusion chromatography allows you to separate by size. So depending on what you want to separate on, you can find a chromatography column that will allow you to separate based on those techniques. Now, what are the differences between this and electrophoresis? Well, electrophoresis uses an electric field to push the molecules through the sieving matrix. Chromatography uses high pressure. It's referred to as HPLC. HP stands for high performance. It also stands for high pressure liquid chromatography. Because this column is packed with a slurry or packed beads, it takes a lot of pressure to move the liquid sample through the column. That's why you need a high pressure pump in order for this to work. So we talked about the stationary phase. The column is eluted with a mobile phase. So after you load your sample like this, you have to add your solvent after that, and the solvent will push the molecules through the column, and the molecules will go through at different speeds. And then you can just collect the separated compounds as they elute from the column. So a typical system would look something like this, where you have these high pressure pumps. Remember, you need a very high pressure to drive the liquid through the column. This whole portion here is an injector. The injector is basically responsible for just adding a tiny bit of sample at the top of the chromatography column. This is just a valve that allows a little bit of a sample to come in here and go into the column. After the sample is loaded, then you have to push in the solvent, or the mobile phase. So after the solvent is inserted just into the top of this column, then they push in the mobile phase through here. And that's what pushes the molecules through the column. The molecules come out at different rates. If you want, you can collect them. So that's what this fraction collector is. Before the fraction collector, there's some sort of detector. And the detector is monitoring the chemicals that come out of the column. Okay, usually it's based on some type of light signal. As you know, molecules absorb light. So if you shine light into a certain area of the channel, when the proteins are passing through there or some other molecules are passing through there, they will absorb some of the light. That changes the light signal. So that's an absorbance detector. There's other detectors as well. But the detector is what generates this chromatogram. Time on the x-axis, and then the molecular concentration on the y-axis. You'll get a series of peaks like this. Okay, and each one of those peaks represents a different component one of these components that came out the sample. Any questions on this, how chromatography works? So chromatography is one of those things where people have tried to miniaturize it on a chip, but there are a lot of challenges associated with that. One of the big challenges is that it requires high pressure. Because it requires high pressure, it's a lot of the chips, microfluidic chips, are not designed to handle high pressure. For example, if you're using a chip that's made up of a plastic or polymer like PDMS and elastomer, those things really aren't designed for high pressure operation. They'll break, the seals will break, and so on. But nonetheless, if you have harder materials like silicon, you can do that if the sealing is very good, if you're able to seal your chip very well. Okay, so that's some of the challenges. And then also, generating a very high pressure is another challenge for chips. Micropumps, we've talked about micropumps in the last module. The only type of micropump that can generate significant amounts of pressure is the electroosmotic pump. Okay. And in fact, one of the examples of liquid chromatography on chip, that in fact they did use an electroosmotic pump to do that. So on-chip LC is a challenge because high pressures are required and they can cause chips to break. There's two types of liquid chromatography on chip. In chip LC, external pumps are used. In chip EC, called electrochromatography, electroosmotic pumps are used. So there's two important parts. If you want to design a chromatography system, you have to have a high pressure pump, and you also have to have your stationary phase. The stationary phase is like, I mentioned, it could be a slurry, it could be a packed bed of beads, anything with a high surface area to volume ratio and small pores between them. So, the slurry or the stationary phase acts as a sieving matrix. The different molecules travel through at different speeds. One thing you can do with microfabricated structures is that you can actually design, using photolithography, you can design different structures that act as your stationary phase. You can have beads or porous materials packed into it. This is an example where they use photolithography to define glass pillar structures. This is an example of silicon pillars, and they're made porous by treating the surface with an electrochemical etch. So imagine a molecule going through here. The molecule may interact, because there's so many pillars here, there's a high probability that the molecule will bump into one of the walls and interact with the wall. So let's say you have a hydrophilic protein, and then you have a porous silicon surface, which is also hydrophilic. There's going to be some interaction there, and that's going to slow the molecule down. Hydrophobic molecule would actually travel through this thing faster than the hydrophilic one. So if the surface of these pillars are hydrophobic, then the opposite happens. So by making these pillar structures and treating them with a certain surface property, you can get different molecules to go through at different speeds. You can have pillars. You can also use carbon nanotubes like this. You can use you can buy the slurries commercially, and you can push them into the chip yourself, although that's not always easy to do. You can create nanoparticle formulations in there so that you get a sort of a porous structure. Anything that has small pores in it, anything that has, and if those pores are well defined and have well defined surface properties, then you can use that as a stationary phase, depending on what you're trying to separate. Silica beads here. This is an example of what the chip looks like. There's an electroosmotic pump here. So this part is not the separation column. This is just an electroosmotic pump. And this channel is filled up with glass beads. So if you remember, the reason why you want glass, for electroosmotic flow, you need to have a glass surface, right? Because that glass surface gets negatively charged. So by filling this channel with glass beads, this becomes the electroosmotic pump. When you place a voltage across here, you'll get flow going from up to down. This is the separation column down here, and this was made using something called a sol gel. And so the separation happens here. The pump is in this portion of the chip. And it looks like they're disconnected. There's like something in the middle here disconnecting the two. But they actually are connected. This is just a region where there's a shallow channel. The shallow channel prevents those beads from going into the main separation region. By placing a voltage between this inlet and outlet here, you can get a pumping effect going down like this. And it turns out that will also drive some liquid into driver sample, which is here. It'll drive that into the column. These are examples of separations that one can get using the chip like this. We have just a few more minutes. I just want to finish up this quickly so that we can go on to particle separations in the next class period. Just a couple more topics on molecular separations. This particular technique, it separates DNA molecules using a pillar array. Now, I apologize for the confusion. This is going back to this is no longer chromatography. This is using electric fields again. But I just want to show that you can separate molecules. You can separate molecules based on by using a pillar array instead of a gel. This is microfabricated. This is an interesting concept. They call it a DNA prism, but basically it's an array of pillars in a microfabricated device. These pillars might be, 10 to 100 microns in size, and the spacing is roughly on the same dimension. They do a pulsed electric field. They do an electric field in the diagonal direction and then in the horizontal direction. And it turns out that the large molecules travel more slowly due to interactions with the post array. So this red line here shows a long DNA molecule. The blue line shows a short DNA molecule. When you apply the electric field like this, and the electric field is applied in the direction of parallel to one of the lines, one of the row of pillars like this. So both molecules are traveling through here. They're not impeded by the pillars. However, when you apply the orthogonal field, this DNA molecule is forced to change direction. Okay, and you can see that it forms kind of like a hairpin structure like this, or like a 60-degree structure. The small molecule can the small molecule can respond to this electric field easier because there's less drag exerted on the molecule. So the small molecule can travel through these pillar arrays faster. So as a result, if you keep on pulsing E1 and E2, going back and forth between the two, the small molecule DNA will actually end up moving away from the large molecule DNA, moving away in the horizontal direction. So by doing this, you can separate DNA fragments very, very rapidly, like less than 15 seconds. Again, traditional gel electrophoresis requires hours. The downside of this is the separation resolution is sort of low. It's limited to large DNA molecules, but it nicely demonstrates that you can use microfabricated components as a stationary phase, as a sieving matrix, whatever you want to call it. This is another example of a work where they used microfabricated and nanofabricated structures. Here, in this work, they were actually using very small gaps. Okay, so the spacing between these pillars might be 100 microns or so. In this work, they're actually using gaps that are 100 nanometers. So they're about 1,000 times smaller. Now, when you make gaps this small, these themselves can serve as pores because they're approaching the same size as molecules. In some cases, they're even smaller than the molecules. So this is the key difference between this previous work and this work that was done by Jong-Yoon Han's group at MIT. They published a well-known paper on this. So there's a few interesting things that happen, and this will end with today. First of all, this is the nanogap. Okay, there's a pillar structure here, and there's a cap on top of it. And between those two structures, there's just a 100 nanometer gap between them. There are microfabrication techniques, nanofabrication techniques that you can use to make such gaps and to make them relatively reliably. So there's three effects that can happen. So you apply a voltage across from one side of this pore to the other. So the negative here and positive here. So one possible thing that can happen is called oxygen sieving. The molecules are smaller than the pore. When both molecules are smaller than the pore, the small molecules travel faster through the pore than the large ones. This is a relatively simple idea to understand. Small molecules will travel faster through the pore. This is similar to just traditional gel electrophoresis. Now, if the pore is really, really tiny, if both molecules are larger than the pore, then the larger molecule might travel faster due to entropic effects. Entropic effects you can think about like, in this case you can see that both molecules are physically larger than the pore. In order to get through there, this protein molecule may have to unravel, go through the pore, and then acquire its original shape again. All right, so when you get into this regime where the pore size is smaller than the molecule, then there's entropic effects involved. And then the final mechanism that they talk about is when the molecules are about the same size as the pore, the interactions with the Debye layer make the charged particles less likely to enter. So Remember, the Debye layer is negatively, the Debye layer is positively charged, but the layer right next to it, the Debye layer in the solution is positively charged, but the wall is negatively charged. So the fact that the wall has a negative charge is going to prevent negatively charged particles from entering just due to Coulombic repulsion. So you imagine that there's a bunch of negative charges along the wall of this orange wall that you see here. So that's all negatively charged. Negatively charged particles are not going to want to go through there because they're going to be repelled like charges repel. So that's electrostatic sieving. So based on these different mechanisms, they do something similar. They have an array of these structures. They apply a voltage in the y direction to make all the molecules go down. And they also apply a voltage in the x direction to get these molecules to go through these nanoscale structures. And depending on which one of these operational modes you're in, the different molecules will kind of separate out into different channels in time. And you can't see the individual structures here because they're too small. But what happens is that over time, you inject your sample onto one side, they start traveling out at different speeds. This looks kind of like continuous flow electrophoresis. But the separation resolution is quite good. All right, you see the different streaks representing different size molecules. So a 2D array of such gap structures can be used to separate particles depending on these three mechanisms. So in this diagram here, these the light regions represent these are deep channels. The dark ones are the pillar, and the gray regions are these the nanoscale structures. So in order for this red molecule to traverse through the nanoscale structure, it has to undergo one of these sieving mechanisms. So over time, the molecules that's able to make it through those nanoscale structures will move to the right, while the molecules less likely will be more straight like this. So interesting technique. Because we're out of time today, we're just going to skip over the gas chromatography. I added this today. It's not in your earlier version of the notes, but I'll just mention, just for information's sake, that gas chromatography is another technique which is used to separate gases in the gas phase, and that's traditionally done with a large instrument, but there's been a lot of recent effort in the last 10 years or so, and this was done a lot at the university that I was at when I was a grad student, University of Michigan, where they miniaturized gas chromatography systems, and they've gotten to the point now where they can detect complex mixtures of things like explosives. They've done exhaled breath analysis to diagnose tuberculosis. Very nice, interesting systems where they separate out a mixture of gases using a long column, so similar to chromatography, but this is in the gas phase instead. Gases separate out in time, and you get these chromatograms. This is a big area of research in the MEMS community. So sorry that we went over today. We'll pick it up on Monday's lecture. We'll start talking about particle separations, separating different sized particles in flow.